Okay, today we have a Zeiss Axioplan 2ie imaging upright microscope. We have this set up for DIC. It's a transmitted light DIC. The light is coming through the base, going through the condenser, through the specimen, and into the objective. Okay, we have it currently with a DIC image. We have a Plan-Neofluar 40x dry objective lens. Okay, let's get it in focus. Let's move the slide around. This is a live blood cell image. We just took the blood specimen a little while ago. You can see the red blood cells. Now I'm going to change the shear plane on the DIC prism. I rotate this knob. If you notice, we're going through like a pseudo dark field at this point, and we keep rotating it to the other side of it. This just affects our contrast and how we look at it. And I'm going to turn it the other way. I'm going to go past that pseudo dark field, which is right in here. It's a pseudo dark field. Now I'm on the other side of it. You can see the red blood cells. You can see how they're indented. Here's one even moving, just slightly moving. So we know it's live because they're moving. More of them in here are moving. Now let's take a look at what bright field would do. I'm going to disengage my analyzer. That's the bright field image. As you see, I've got a very significant loss of contrast. I can see the red blood cells, but I don't see them very well. That's bright field because I've disengaged my analyzer. Now I will re-engage the analyzer. Here's the DIC again. It's a much better image. Now, this is a 40 power Plan-Neofluar objective. Let's take a look at a 20 power. There's the 20 power. And let's change the shear plane again. Here's our pseudo dark field. Take it to the other side. We're getting some very good contrast. Turn it the other way. Our pseudo dark field again, now on the opposite side. All this is giving me some very good contrast. There's a lot of adjustments I can do. I can adjust the iris in the condenser. I can adjust the screw on the DIC prism. I can, of course, adjust the focusing. All of this will change the image that I'm getting. At this point, I'm pretty satisfied with my image for the 20 power. Now let's change it to a 10 power. This is a Plan-Neofluar 10 power Zeiss objective with the Zeiss DIC prism. Everything else is the same. We have a prism inside the condenser. Now if we look at this, these red blood cells under the 10 power, they're hard to see. They're very tiny on the image. So it does help to have a higher power magnification, as we can see, for live blood. The 10 power is just not enough. Let's go back to the 20 power. By the way, let's take this back to bright field for comparison once again. There's a bright field. I can try focusing it, but it doesn't really help me. It was already in focus. I just don't have enough contrast with straight bright field. Turn it back to DIC. I have some very good contrast. I can see some very good detail. And we will change it to the 40 power again. We can see the red blood cells very well. Okay, that's it.